So, hello everybody. Thanks for joining us today in our MTL lecture series. Today we have our new fellow Oskar Staufer with us. Hello, Oskar.
Hello.
Nice to have you here. He is at the University of Saarbrücken and the Leibniz Institute in Saarbrücken, and yeah, he will give today the talk about the research he's doing within the Matter to Life. Before we start with the scientific talk, I just want to briefly explain you the system we have. So this is a webinar. You cannot switch on your sound or your camera, but you can ask questions by raising hands, or there's an FAQ section somewhere at the bottom of your screen where you can type in. So it says Q&A or FAA, and you can type in questions already during the talk, and you can also upvote them in case you're more interested in a question somebody else asked. Then it will get more to the top of our list and will be asked the first. So after Oskar has finished his talk, so please use this option to ask any questions. So before we now dive into your research, Oskar, I want to ask you a few questions. So maybe the first question would be, why did you join the Matter to Life program?
Mm, I guess because it's the program in Germany to pursue the type of research that we're doing. Yeah, there's a big school of thought of people that are interested in physics and biology, but also in engineering biology and reducing the complexity of biology. And I think it's very rare that you find so many people with a similar mindset that pursues this type of approach to do research.
Okay. And how do you think do your research fits into the interdisciplinarity research yeah, field of Matter to Life?
I think I'm a little bit biased when it comes to say how it fits. The only thing I can report is essentially when I meet with the students, usually the beginning of the week, Mondays and Tuesdays, there is a day or a typical day would be talking in the morning about the physics of membranes. Then the next session is about T-cell immunology and how patient data fit to what we observe in the lab. And the next thing that we think about is a new clinical study that arrived. And then we go back to the physics of cell migration. So uh, just from the type of research that we discussed, I think we are highly interdisciplinary, and I hope you will see that in the talk today as well.
It already became clear listing the institute and disciplines, which is like, okay, then different institute there, different total different discipline there. Okay, great. And if you wouldn't be a researcher, I mean, no, you are one, but what would you be your alternative career path you would have taken
I
or
honestly,
tried?
I don't really know. Yeah, the only thing I can say is it would be probably something that comes with a lot of freedom. I guess that's what I enjoy mostly about science, that you can pursue own ideas, and of course, sometimes you need to ask other people for money to pursue these ideas and for the room to actually perform the type of research that we do. But at the end, the core of what I like is actually following up with a team of people things that we just find interesting at the end of the day, yeah, and that we think could have a value in the world out there. So it would be any job related to that. Maybe that's a good description of being self-employed somehow, yeah.
Yeah, any kind of topic. Asking people for money and do whatever you want. Sounds good to me. Okay, so thank you for introducing yourself. And then I would give the word to you, and I'm happy to learn a bit more about your research.
Thank you very much. So, I'm actually super happy to present today as a fellow, because some of you might know that I actually my first contact point with the Meta to Life was a very different one. Yeah, I actually started as a student in the Meta to Life school, and for me, this is why this is kind of an incredible opportunity to reconnect with the whole community, to also kind of give back a little bit the things that I received from the school, maybe also integrate the student view a little bit among the fellows, because not so long ago I've been a student myself. Yeah, so I'm really happy to present today. And um, I thought a while about what to present, and I guess the decision I took was a decision that is kind of in between the things that we do at the end and that are important for the school. So things that I will be presenting about are of course, on synthetic cells, that's what we work on, and what also a great part of the school is focused on. But I also wanted to give a little bit of a different touch of what we do with these synthetic cells. You will be seeing some results in research that look in the physics of immune cells and how synthetic cells could help us actually to understand the physics of immunity a little bit better. But you will also see a great part of the work is devoted to improve therapies. And I think this is where synthetic cells are actually quite powerful for us. Yeah. So I will be showing you some things where we use these advantages of synthetic cells, meaning their controllability and scalability, that contrast very, very strong to natural cells, and essentially how we use that to at least give a glimpse of how to improve immunotherapies in the future. And the specific type of immunotherapy that I will be talking about in this talk is adaptive T cell therapy or adaptive immunotherapy. And for those people that are not fully familiar with this process, I want to just give the very, in a nutshell view of how T cell immunotherapy works. The general concept is that we apply this type of therapy to cancer patients. There's also more and more approaches towards autoimmunity diseases, but I will be talking mostly about cancer patients. And these cancer patients can easily donate some blood. Out of this blood, it is standard routine in the last 80 years probably to isolate immune cells, specifically T cells out of these, and expand them in culture. So under ex vivo conditions, just in the incubator. And these T cells after expansion, let me put on my pointer here. These T cells after expansion can then be genetically re-engineered to give them a new ability. And that new ability is essentially identifying cancer cells, targeting cancer cells, and killing cancer cells. Yeah. It's a really beautiful approach. Yeah. You can take blood from a patient that suffers from a cancerous disease, train these immune cells outside of his or her body, and reinfuse them back into the patients with their new genetic learning ability to identify cancer cells and then kill them. And there's several therapies where this really works very well. Unfortunately, only about 30% of the patients really have a pronounced response, a sustainable response, and can be cured with such type of an immunotherapy from their cancerous disease. And that leaves us with 70% of the patients that essentially don't profit from that. Yeah? And we see it as a little bit of our mission in the group to not reinvent the whole process, but essentially apply synthetic cells in a way that we can improve the outcome of these 70% of the patients. Most people in the field would actually work exactly on the step 3 here. This is where most of the cells of the field of synthetic immunology is going into, essentially trying to re-engineer genetic variants that can be introduced into these T cells to either recognize the cancer more efficiently, kill the cancer more efficiently, have different triggers to identify the cancer, and so forth. Yeah. However, we think there's another handle that is very critical for the efficacy of this therapy that has remained untouched in a large part. And that's actually step 2 here. So if you think about cells and their genetic made up of a hardware and a software, yeah, I would say people focus on the software a lot, but I think the hardware in which the software is introduced could still be improved and thereby improve the outcome of patients in here. And I want to tell you how synthetic cells could be a way to improve the step number 2 here in this overall process. When I speak about synthetic cells, I think one special thing that I will be presenting in this talk that you might have not seen so far in many of the in a lot of the relevant literature is actually that we don't stop working with single synthetic cells. And the story that I want to present you is a story of how to program self-assembly capabilities into synthetic cells and form fully synthetic tissues out of them that are structured, that are organized, and that can be tuned in their biochemical and mechanical properties, all targeted to improve step 2 in here. So I cannot tell you the story without going a little bit deeper into immunity and how immune cells actually work. I don't want to talk about the whole immune system. I will be focusing on T cells. And I want to talk a little bit about the places that T cells feel probably at home. And those are your lymph nodes. The lymphatic system is a quite complex system. You have many lymphatic organs. You might know the spleen, which is probably the first one that comes up to. But all scattered throughout your body, you have these tiny microanatomical structures that we call lymph nodes. And the lymph nodes are the places where T cells usually go to to understand how their host, so the body or the patient, the person behind them, is actually doing. That's the places where signals and information is integrated for immunity. T cells go there to understand if there's any antigen, any pathogen, any cancer cell that they should be reacting to is around in the patient. Yeah. And maybe one thing that you have noticed when receiving a vaccine or also when suffering from any kind of an infection is these lymph nodes swell. And the swelling process is mostly attributed to the fact that these T cells, and some other cells as well, but let's talk about T cells, actually start expanding. Once they receive the trigger, once they understand or get the information, there's something that you should be reacting to, they will start proliferating a lot. And essentially, this is the process that adaptive immunotherapy tries to mimic in cell culture. Yeah. So what we're actually trying to do in the cell culture is mimic a lymph node. Yeah. Whenever you want to expand a T cell, you need to trigger it to make that T cell think it has been communicated to from the host's body to actually start and react against a specific antigen. And that swelling, however, we think is also crucial to navigate the process of the activation. Because if you don't regulate the way this immune system and T cells are activated, you get something that we call autoimmunity. So your body actually invests a great amount of energy to regulate immunity. And typically, people will look at the biochemistry side of this regulation. How do specific inhibitory signals, immune checkpoint inhibitors, and so on actually inhibit the activation of T cells, or at least regulate their activation so you don't have an overshooting immune system. What we think, and also what some papers report, is that mechanics also play an important role in this regulation aspect. Yeah, and I will make the point later that the mechanical aspect is absolutely not integrated in the process of adaptive immunotherapy. So if you want to improve adaptive immunotherapy, it's not only worth understanding in engineering the biochemical side, but also integrating a biomechanical side into this. And the reason I'm putting up this image here, it's pretty clear because the mechanics, so the stiffening, will also control how fast and how strong these T cells can actually proliferate. And that's something that I will be talking a lot about in the talk. Okay, let's have a look at how T cell expansion is actually done in these therapeutic settings. I want to highlight this is not something that just happens in cell culture, but is actually an approved GMP compatible procedure for the actual production of a therapeutic compound. The state of the art to do that is actually take some of these T cells in culture and incubate them with simple polystyrene beads. These polystyrene beads will have antibodies on the surface that are able to bind to the T cells and activate them. When I play this video here, I hope I'm able to play it. Yes. You see a time lapse of this process. Yeah. So individual T cells will room through the cell culture plate, find individual or singles of these polystyrene beads, bind to them, and then start a proliferation process. On the molecular level, it looks roughly like this. This is a polystyrene bead, magnetic, so you can remove it afterwards after the activation process happened. And it has two types of antibodies on the surface. It's an antibody against CD3, that's a part of the T cell receptor, and a co-stimulatory antibody against CD28 on the surface of T cells. This, together with another cytokine that is added, interleukin 2, is usually enough to activate a T cell. And now maybe you can understand a little bit why, after doing a PhD in synthetic cell engineering in the Matter to Life, I then decided to go into immunology, because I think this process is already quite a good example of reduction of complexity. You have a whole immune system that is about regulating the activation of immunity and making sure things are activated only when they should be activated. And actually, by integrating 3 different proteins in cell culture, you can achieve almost the same process. I'm saying almost, there's a nuance and difference to it, but a large part of the phenotype can be reproduced with only 3 proteins presented or activated in cell culture. A beautiful example of reduction of complexity. But if we think about how this thing works in the lymph nodes, maybe there are some other signals we would like to integrate in future into this. In vivo, the actual process is much different. In vivo, we don't have a polystyrene bead. We have an antigen-presenting cell. It's mostly a dendritic cell. And it doesn't have antibodies on the surface, but actually major histocompatibility complexes that bind to the T cell receptor, and not a co-stimulatory antibody, but a full ligand that we'll find, for instance, an integrin receptor on the surface of the T cells. Additionally, there's a whole cytoskeleton and mechanical sensing component in these T cells that will also integrate the signals on a mechanical level provided through these ligands. There's cytokines exchanged between both of these cell types. There's ligand mobility that again impacts the clustering on the surface of these cells, the protein clustering at the interface. There's an extracellular matrix in which all of this happens, and there's multiple cell types around that will all contribute to this. So you see, the process is extremely complex. There's many things that you need to integrate, and all of these will play a role. So it's important to understand which processes shall we integrate if we want to build something like a real lymph node for our incubator, and which processes are maybe less important. Fortunately, there's a lot of literature that we as synthetic cell engineers can rely to, and now it's mostly our job and our engineering challenge to integrate all these processes in a way that they're physiologically relevant, or at least that they happen in the physiologically relevant time and spatial scales. So what we do, I already took that a little bit forward, is we build synthetic but programmable synthetic lymphatic tissues, or synthetic lymph nodes. Yeah? On the left side, you see a histology image of a real lymph node. On the right side, you see one of our synthetic lymph nodes. And synthetic here means fully synthetic. There's no living cell at this stage in these lymph nodes. They're all made out of synthetic cells that bind to each other and form a real tissue-like structure. And what I want to tell you for the next couple of slides is how you can build from individual synthetic cells such a synthetic tissue structure. And then I will move in a little bit into how you can use that for real T cells extracted from humans for their activation in a therapeutically relevant context. First, let me tell you a little bit what synthetic cells mean in our context. You might know there's tons of different types of synthetic cells around: GUV, colloidosomes, coacervates, polymerosomes, and so on. And we didn't start from picking a specific model, but we started from the idea of we want to have specific functions integrated. First of all, we want to have a lipid membrane, and I will show you why that is important. When I have control about the mechanical properties, so the stiffness of the synthetic cell, we want to present ligands in a very defined manner on its surface, and in the best case, it should be able to secrete cytokines and metabolites at the same time. And putting these four attributes together made us realize there is no synthetic cell model out there that can combine all of these. So we had to come up with our own one, and I will show you what that synthetic cell model is like. So what we are building is something similar to a bead, so a particle-like structure that should mimic a real antigen-presenting cell. This is an electron microscopy view where you can see real T cells binding to the surface of these dynabeads. And what we try to build is something very similar, but different on the membrane and the mechanical side. So what we are producing is droplet-supported lipid bilayers. I will show you an image of the molecular makeup of these lipid bilayers in a second. The important thing is these should be brought in culture. So under culture conditions, high ionic strength, a lot of protein around 37 degrees for several days in contact with T cells. Yeah? So stability is a very important point to consider here. So we integrated a membrane because because immunology is about membranes. I showed you this electron microscopy video to make the point that immunology and immunity happens at membrane-membrane interfaces. So if you want to build a synthetic cell model to activate immune cells, it should have, in the best case, in a membrane. Yeah. So what we use is a standard lipid bilayer that we coat on the surface of droplets. You see a Frapp experiment in here that essentially shows you the lipid membrane of these synthetic cells is mobile. We can then also tweak it a little bit to have a specific membrane topology. So for instance, to have microvilli structures that present proteins on the surface. Thinking about the stiffness of these synthetic cells, I will show you in a second a video here that highlights the relevance of stiffness in immunity. It's a video where you see not a T cell, but a natural killer cell in culture with cancer cells. And that natural killer cell will swarm around, identify these cancer cells, and trying to kill them. And when I play the video, please have a look at the deformations that happen on both cells. So on the target cell, the cancer cell, and the immune cell. This video, by the way, is not from us. This video is from our colleague Markus Hoth in Hamburg. So I think one of the most interesting videos for the mechanics of immunity that I want to share with you. And I'm playing it here now. You see the cancer cells in orange and the natural killer cell in the bright field image. The killing cells turn green once they're dead, and you see how much this immune cell starts pushing into their target cells. Yeah. So if you want to build a synthetic antigen presenting cell, you need to make sure that it's deformable. And I think that's on the synthetic cell model side, the major difference that we have to these polystyrene beads. These are very rigid beads. What we use are oil droplets. Yeah? And these oil droplets, of course, are deformable if we look at them through real-time deformation cytometry. We've also recently done some micropipette aspiration studies on these. They're definitely deformable, and most importantly, their elastic modulus is roughly in the range of what you would expect a natural antigen presenting cell to have. Yeah. So we have a lipid membrane and a mechanical support that matches roughly the mechanics of real cells. Now, most importantly, we want to put membrane proteins on the surface. I said immunity happens at membrane and interfaces, so we need something like an antigen, maybe something like an antibody against CD3, CD28, what I showed you in the beginning. And the easiest way we can do this, and the most controlled way of doing that, is to use functionalized lipids that we put into the membrane. That's mostly NTA functionalized head group functionalized lipids that we then can couple his-tag proteins on the surface. You see a GFP signal here on the membrane of this synthetic cell model. And then we've also recently started to integrate synthetic cells that can release compounds, mostly metabolites. We're also working on some cytokines from the inner core of these synthetic cells. You see here a fluorophore integrated in such a synthetic cell model. The trick here is to produce double emulsion droplets, so essentially a water and oil in water system that integrates whatever compound should be secreted in the aqueous core. And then what we do is we engineer the surfactant layers that stabilize the inner core droplet to the outer core droplet, so that there's a micelle transport process happening between the inner and outer interface. And by this micelle transport process, you can also secrete small molecular compounds from the inner core. By then essentially adjusting the surfactant concentration that is stabilizing the inner core, you can also adjust the release kinetics of this whole process. So that's the synthetic cell model that we're using. We call them droplet-supported lipid bilayers. Inner core oil droplet surrounded by a lipid bilayer that we can put proteins on the surface. What I tell you now is not too much on the synthetic cell characterization and how they really interact with different immune cells on the molecular level. I want to highlight more how we can take these synthetic cells dispersed and program them in a way to form a complete synthetic lymphatic tissue. So you actually see them by eye. When you look in our incubators, you would see these synthetic lymphatic lymph nodes by eye. Yeah? These are macrostructures. And then in the third part, how we apply these for application-based purposes in the realm of adoptive cell therapy. So this is another groundbreaking video from ours. It's actually one that I took very early on in my postdoc days, which kind of exemplifies the breakthrough of how we were able to build these complete macroscale tissue-like structures. What you will see is time lapse, roughly 18 hours, of individual dispersed synthetic cells. That's the nebula of droplets that you see swirling around in this video, and how over time they start to find each other and aggregate and then form essentially a tissue-like structure. Yeah? So pay attention to the scale bar. This is real macroscopic imaging here. And over the process of the self-assembly, they start binding to each other and forming larger scale objects. Larger scale, we typically work in the millimeter range, 1 to 2 millimeters. That's also roughly the size of a healthy lymph node. Once they get inflamed and T cells start to expand, they can reach several centimeters in size, of course. But we can actually also build larger ones just for the sake of parallelization. We build smaller structures in here. How do we do that on a molecular level? The easiest way of doing this is using a simple protein that will interconnect individual synthetic cells with each other. Such a protein could be streptavidin. If you then integrate biotinylated lipids into the lipid membrane of these synthetic cells, streptavidin is multivalent, can bind several of these biotin residues on the lipid membrane in each synthetic cell, and then cross-link essentially individual synthetic cells. That's the easiest, most robust process to do that. You can tune the tissue mechanics by just adjusting the amount of streptavidin that you integrate or the type of or the number of biotinylated lipids that you integrate. We also recently published an approach where you can use DNA aptamers to also program which synthetic cells should be connecting with what partner in the mixture. All the data that I will be showing you is based on these streptavidin droplets for the sake of stability. And I think this is important to mention here because if we think about the translational potential of synthetic cell technologies, we as synthetic cell engineers, in many cases, underestimate how robust and stable such a system should be built. Yeah? So robust means it needs to work off the shelf immediately. And stability means the culture conditions are quite harsh. Yeah, not only the routine culture, so a high protein or protein-rich medium and high ionic strength buffer environment, but also the cells secrete a lot of metabolites. They secrete proteases that can degrade tissue. That's all the advantage a simple polystyrene particle has, and that we need to compensate with these more advanced synthetic tissue models. Yeah? And here, biotin streptavidin is just a very robust approach to go into and actually have synthetic tissues that are stable over days and weeks in culture. So that's how it looks on the ultrastructural level. If you zoom into one of these synthetic lymph nodes, you see the membrane of these synthetic cells, and they form these tiny intercellular connection spots that are enriched in streptavidin. That's essentially the interface that you want to tune for the or that you want to program for the synthetic tissue assembly. There's three things that we care about in these synthetic tissues: the biochemistry. We mostly adopt things that have been established, the anti-CD3, anti-CD28 antibodies that we put on the structure, and some cytokine mixtures that are well documented. But we mostly engineer the structure and the mechanics. So the biochemistry, I already told you, we just add through a maleimide functionalized lipid head group antibodies to the surface of these synthetic cells so that they're simply able to activate T cells when they're getting contact. This is now the first image that I'm showing you of such a patient-derived T cell infiltrated in such a synthetic tissue in close contact with a synthetic cell. DSLB here is for droplet-supported lipid bilayer. So they bind and scan the surface of these synthetic cells in the search of these antibodies that will make them activate. I want to show you this video here of these synthetic cells navigating through the synthetic tissues. The important thing is, or maybe first I should explain that the T cells are here labeled with their nuclei and cyan. And the synthetic cells tissue you see in magenta in the background, it's not very well resolved. Here the aim was to have a good time resolution, not necessarily spatial resolution, but you can anticipate where the individual synthetic cells lie and how the real T cells then actually migrate through this tissue in the search of antibodies or antigens on the surface of the synthetic cells. And this, of course, is a very important process that you need to mimic because this is how real T cells look for their signal in lymph nodes. Lymph nodes are not a void, empty space where soluble antigens float around. T cells collect information over time through a migration process. Yeah? And we were actually quite happy to see that the T cells can migrate quite steadily and efficiently through these synthetic lymph nodes and scan multiple synthetic cell surfaces over time. And I will show you some migration tracking data later that can tell us at least a little bit about how the physics of this process play a role to optimize the search efficacy of T cells when it comes to activation. Well, after some time, roughly after 4 to 7 days in culture, what you will see is that these T cells will form these proliferation clusters. This is very reminiscent of what you would see in a real lymph node structure, where once a T cell has been activated, it will start to proliferate. This is what the whole tissue expansion is based on. Yeah? You see all of these clusters were usually one T cell that was activated that then started to proliferate and form a cluster out of it. Thinking about clusters, we also need to think about structure. The lymph node is not a homogeneous cell tissue. Yeah. It has specialized regions for all type of different immune cells, for B cells, for T cells, for macrophages, NK cells, and so on. What I showed you so far is essentially a simple aggregate, so kind of a colloidal structure of synthetic cells that just form a isotropic, homogeneous synthetic tissue. But the structure is very important to regulate mechanics. In a real lymph node, you have loosely bound immune cells and lymph node cells that will regulate the expansion process, while you have certain centers that are highly enriched in antigen presenting cells that will be the major drivers for the activation process. So a synthetic model should actually recapitulate both sides, an activation cluster, but also a surrounding scaffolding tissue that allows for an expansion to happen. And this is essentially what we integrated in our synthetic lymph nodes. A T cell expansion zone, that's a zone of synthetic cells that carry the antibodies on the surface that will make the T cells activate, and a more loosely bound scaffold tissue that will allow to form a coherent structure, but also that is expandable over time and regulates the tissue mechanics behind it. Most importantly, if you incubate that with real T cells, again here shown in cyan, the scaffolding synthetic cells are in green and the antigen presenting cells are synthetic cells are in magenta, and you perform a co-localization quantification of where the T cells will go. The good thing is most of them will actually start localizing in the activation zone. So the overall structural assembly works. The T cells don't randomly proliferate through the tissue structure, but they actually go into the activation zones of this whole thing. And now we need to think about the mechanics of this tissue. I already told you by regulating the biotin concentration in the membranes of these individual synthetic cells, we can tune the cross-linking degree. And what we then did is we performed a parallel plate compression analysis, so essentially just pushing on these synthetic tissues to understand how stiff they are with varying concentrations of biotins in the surface. And what we saw is essentially that this can regulate the mechanics of the tissue. Looking at real mouse lymph nodes and their mechanics, we still see we're roughly an order of magnitude away. I just want to highlight that this is not the real tissue mechanics of what we are mimicking here. Lymph nodes have a whole capsule around them made of very strong connective tissue, and that is what has the biggest impact on their mechanical properties in here. These tissue mechanics that we see in here is actually quite representative for the internal structure of a lymph node of many other connective tissues. So now that we integrated the biochemistry, a structure and mechanics, we can start to quantify the behavior of T cells that will migrate into these synthetic lymph nodes. This is a video or intravital time-lapse analysis, so essentially looking into the lymph node of a living mouse and analyzing the migration behavior of individual immune cells. You can see the T cells in red, how they scan such a lymph node surface. And I already showed you the video of how the T cells migrate in our tissues. This is now such a heterotropic tissue with activating zones and non-activating zones, where we can now, based on highly time-resolved time-lapse imaging, quantify the migration behavior of these T cells. We see, just as one example, for instance, some T cells will squeeze through individual synthetic cells, so try to find their way through. There's a mechanical component on this. Some will travel from a scaffolding part into the activation part and back out again if they didn't receive enough signals. So the whole behavior of these T cells is actually quite complex. Just want to give you a glimpse in what we saw so far in this type of analysis. If we compare a dense tissue, so a tissue that have 5% biotin lipids in the membrane, to a very loose tissue, so that's 1% in this case, the migration dynamics of these T cells in 3D varies a lot. So you see individual paths here of T cells in this tissue. But the second thing that you observe when you look a little bit closer is T cells sometimes move, stay around a specific spot, scan the surface or the interface there, and then will move along in somewhere else. Yeah? That is the dwell time that we quantify in order to understand how long T cells actually integrate signals in a specific region. And what we see from these analyses, that T cells will integrate information not in a second time scale when it comes to their migration, but in a minutes time scale, roughly around 60 minutes. And that process is much faster in dense tissues. Yeah, so if you compare these two graphs, you see here, for instance, quite well that the T cell made a pretty fast decision. And this is what tells us the mechanics and the structure of this tissue will influence the search efficacy of individual T cells, and by this also regulate how immunity can choose between an activated T cell and non-activated T cell. And of course, we are now following up with some ideas of whether the mechanics or the swelling of the lymph nodes is what actually drives the stiffening, and what will define if a activation process such as something like a vaccination is actually efficient or not. I want to show you just some more quantitative data on the relevance of structure and mechanics in such a synthetic tissue. We produce two types of synthetic tissues. One that we call heterogeneous, that is, the activation zones are very dense and the scaffolding tissue around is quite loose, but we also reversed it. So we have an activation zone that is quite loose and a dense scaffolding tissue. I'm showing you this because it highlights the power of what we can do with these synthetic tissues that is essentially impossible to do with real tissues. You can start flip-flopping properties of the tissue back and forth that you couldn't do with a real lymph node, and understand the impact of this on the activation of T cells. The way that we measure or quantify activation of T cells is that we look at the CD25 expression on the surface, and we benchmark all of our analysis to dynabeats. Yeah? If you quantify the activation between the heterogeneous tissues, so this structure, and the reverse tissues, with varying concentrations of activating synthetic tissues, so 75 here indicates 75% of the synthetic cells in this tissue are from the activating type, you can see that none of our conditions is actually better in activating T cells. That was on a first glance quite disappointing. We're trying to build up a more advanced technology that could compete with the state of the art in the field and expand T cells much better or more efficient than the simple polystyrene bead with two antibodies on the surface. Well, first of all, I made the point, we've been working on this for 2 years, and dynabeats have been developed for 30 years probably, or something like this. So it would be a shame for them if they don't perform better than we do. But second of all, we thought, well, maybe looking at one signal is not enough. CD25 just quantifies the degree of activation of these T cells. It doesn't tell you anything about the quality of these T cells or what kind of properties these T cells are. So maybe let's look a little bit deeper in what the T cells that we expand are maybe better at than these dynabeats. Yeah? So we quantified many other cytokines that these T cells release, and we got a surprise. We found that all of our synthetic tissues actually produce higher levels of interleukin 10, interleukin 17, and interferon gamma. For most T cells, this is not a surprise. But for the T cells that we use, this was a surprise, because these are CD8 T cells. These are T cells that usually don't have a lot of regulatory properties. They're mostly there to kill. They're there to identify the targets, mostly cancer and viral infected cells, and kill them. You would not expect any secretion of these cytokines. What told us that what we are actually expanding in this synthetic tissue structure is a regulatory type of CD8 T cells. We looked a little bit into the expression values of individual proteins and their signaling activity and phosphorylation state. We were also able to identify why we produce in the synthetic tissue context these type of phenotype without going into the molecular mechanisms behind it. It's mostly mechanoreceptor signaling behind it. But what we found is essentially a synthetic cell technology that we can assemble into a tissue to produce regulatory T cells. And regulatory T cells, I told you in the very beginning, are interesting when you want to apply adoptive T cell therapy, not for cancer patients, for patients that suffer from autoimmunity, where you don't have not enough immune cell activation like in cancer, but where you have an overshooting of activity, such as in autoimmune diseases. You want to apply CAR regulatory T cells. And because we integrated the mechanical component and we mimic the mechanical component in our synthetic cells and synthetic tissues, we're actually able to preferentially expand such a regulatory phenotype with our technology. So with this, I would already like to conclude. I showed you some examples of how you can build an artificial tissue scaffold from these synthetic cells. I should maybe also mention here that this is not where it stops. You can integrate additional synthetic cells in there. We use some LUVs and GUVs that we integrated together with these lymphatic tissues. Just showing you that you can be quite creative and integrate different types of synthetic cells that have different types of advantages for specific functions. Yeah. I showed you that you can program these synthetic cells to produce three-dimensional tissue-like macrostructures through self-assembly. The self-assembly is a very critical point if you think about automation, where you don't want to print something, you don't want to inject something, you don't want to have maybe an organ on chip that you can run maybe 5, 10, 20 of them in parallel, but you really want to screen 2,000 conditions, for instance. Yeah. I showed you, and maybe this I did not highlight enough, that natural cells can be brought into culture with these synthetic cells and these synthetic tissues over days and weeks of culture, that they will infiltrate by themselves in these synthetic tissues and receive biochemical and biomechanical information from the hybrids. And at the end, how we can use these type of synthetic lymph nodes to expand a regulatory T cell phenotype for adaptive immunotherapy. And for this, I would like to thank the whole group that took part in one way or the other in this technology. I would like to highlight Anna here, who has been a major driver of this technology, but also Niels establishing the droplet-supported lipid bilayers. Some students were also involved with this, Tamara and Yumin, and analyzing the data. And there's many other people in the group that now move forward this technology for different type of purposes. I'd also like to thank all the people that helped us in this, and also, of course, the funders of our research for giving us the money to actually pursue the ideas that we think are worthwhile pursuing. Thank you very much.
Thank you, Oskar. That was a great talk. In case you have any question, you can now use the Q&A section to type in your questions. Before there are coming questions, I have a question or understanding questions regarding the assembly of the tissue. So did I get it correct? So you cultured or you produce different synthetic cells with streptavidin ratio, and then you just put them together all in once, and they self-assemble into the stiffer. So depending on the streptavidin content into stiffer and loose regions. But how do
Correct.
you then, okay, how do you then make sure that they have this reverse, because then
Yeah.
you should have right islands of more dense and more loose tissue, but not necessarily reverse and
Yeah.
heterogeneous structures.
Yeah.
How do you make that?
Yeah, so there's two approaches that you can take. The one I only briefly talked about is if you avoid the streptavidin biotin, you can use DNA aptamers that have specific complementarity. So you can
Mm,
produce cell type A that binds to cell type B, but not C, but C binds to D, and so on. Yeah.
mhm,
When you want to use only one type of linker, you need to do a step-by-step assembly process. So
mhm,
you first choose, let's say, a self-assembly conditions that will only produce the core, so the activating zones that are very dense, yeah, but you build them in a way that they're very tiny, or you rupture big ones apart after the formation. And then in the next step, you add your scaffolding synthetic cells that also bind to a lower degree, however, with the streptavidin biotin interaction. So you form the scaffolding structure around these, I want to call them for this sake here, nuclei of activating zones. And that's how you introduce hierarchy. You can do that for multiple steps, then break these apart again, add another layer and so on. Becomes quite tedious after some time.
mm,
If you really want to build large hierarchical and structured tissues, I would go for the DNA-based assembly. If
mm,
everything that you need is two different types of structures, then the streptavidin biotin is actually working quite well. And it's also
mm,
an easier bridge if you want to integrate other types of synthetic cell models, right? Because the biotin is a
mm.
quite available chemical moiety, small molecular weight that you can easily put on the surface of other coacervates, GUVs, whatsoever. Yeah.
Yeah, and in the end, it doesn't really, I mean, it's nice if it self-assembles, but it doesn't really matter because it activates anyway your T cells, right?
It does.
Yeah.
So I didn't talk about this because I'm
Mm,
not conclusive at the moment. But of course, if you want to start to apply these synthetic tissues for understanding molecular mechanisms, maybe screening different types of cytokines that might be relevant, but also if you want to screen drugs that might interfere or promote the activation process. We're not talking about the 96 well plate or 300 samples anymore. We need to test thousands of conditions. And there
Mm.
you want to have a self-assembly process. Yeah, you want to pipe
Yes.
things into wells and then physics does the rest. Yeah, you don't want to have people actually handling these samples.
Yeah. I can see you standing like pipetting the hands off. Okay, great. So we have now also the first questions from the audience coming in. So I will read them out loud and then you can answer them just yeah, uh, by telling us. So the first participant said, "Thank you very much for your talk. I was wondering in your migration trajectories, sorry, you could see some form of immune synapse forming between the APCs and the T cell in your lymph node. Or more general, if you think the formation of a synthetic immune synapse will be important for the synthetic version of the immune response activation overall."
Hmm. It's hard to give a yes or no answer to the second part of the question. So we know that these polystyrene beads are actually, if you just take the very same antibodies and coat them on a polystyrene plate, they will activate T cells. So you don't need a fully proper immune synapse that is highly organized formed in order to activate a T cell. Yeah, but of course, being closer to the natural immune synapse, meaning having lateral ligand mobility, cluster formation of individual T cell receptors, segregation of immune stimulatory and inhibitory components on the T cell side, all of these will increase the physiological relevance of the immune synapse that you form. So I cannot say if it's better or worse. We just know that it is important for some processes to actually introduce that for actually to discriminate individual activation signals. What we observe in our synthetic cell T cell interfaces is what I would call an immune synapse reminiscent structure. We haven't performed any highly spatially resolved analysis of where, you know, the different integrins and CD45 and so on are actually located at an interface, but there's an acting organized structure directly at that interface.
Okay, thanks a lot. So we have another question. Thanks for your talk. How do you tune the ratio of CD3 to CD28 ligands on the beads, and how does the ratio control the strength of the T cell stimulation?
So I briefly mentioned that we use melamide to couple the antibodies in the surface. Melamide unspecifically binds both types of antibodies, so you can easily just vary the ratio that you add to these synthetic cells to tune the ratio that will actually end up on the surface of the synthetic cells. Yeah. Now, the second part of the question, how important is it? Not only our group, but also many other papers try to vary this ratio and understand what's the optimal ratio that you need to apply in order to get a good expansion process. So mostly a most efficient proliferation process. And the typical values are 1 to 3, 1 to 4, 1 to 5, something like this. So you always want to have a little bit more of the CD28 on the surface than the actual CD3. Otherwise, you get a too strong T cell receptor stimulation. We go with the very same. We apply 1 to 3 in our approaches. Yeah? We performed some analysis on variation of these of this ratio. Honestly, we never saw a big difference. And I think the major reason for that is the lateral ligand mobility.
Mm.
The literature data is mostly on fixed ligands. Yeah? So there, of course, it matters more if a T cell cannot rearrange the interface as it pleases. In our case, we don't see much difference in the 1 to 2, 1 to 3, 1 to 5 ratio.
Okay, thank you. Just me hopping on. So if you're talking about ligand mobility, do you also see that from if you're looking at your tissue and you have the dense part and the less dense part, that over the time you have lipid exchange between the GOVs and like I don't know about what time frame I'm thinking even, but then get a more normalized distribution of one synthetic cell type?
Yeah. I mean, it's hard to quantify this for all components of the system. The only thing that we can observe are the fluorescent lipids that we integrate
Mm.
to the synthetic cell membrane. And if you look at those after days and weeks, you don't see any exchange. Yeah? So
Okay.
these lipid membranes don't really mix. However, what we see, the T cells and the lipid membranes mix. So once you get the T
Mm.
cells out, you will find lipid membrane signal in the T cells. Why is that?
It's eating up.
Um, Exactly. T cell nibbling is what we call phagocytosis. Yeah, that's a process mostly known from the innate immune system and macrophages. Well, the cell will pick up tiny portions of the membrane, and that is actually a very important process that again regulates immunity behind it because it prolongs the exposure of the antigen, now not on an extracellular basis, but an intracellular basis. Yeah. And we actually have some indication that this is also happening with our model. We don't know if it's good or bad at the moment, yeah? If it helps the processes that we're observing or if it inhibits a more efficient activation on the CD25 level as compared to the dynabeats. The only thing I can say, regulatory bodies don't like that. Yeah, so if you want to apply that to patients, the beauty of a magnetic bead is you can very efficiently remove it. If anything in the final medical product that you put up back into the patient has something of our synthetic cell system, it's not a neck breaker at the end, but it's something that needs to be characterized. And that's I'm just mentioning this because you asked me in the beginning about the interdisciplinary aspects. We think about lateral lipid mobility
Yeah.
as we think about regulatory authorities and what they want to see and what they don't want to see.
Yeah. I can imagine it's gonna be a bureaucratic nightmare. Not only the analysis, but then all the things that follow. Okay, then I have another comment, which is just no questions, just wow. Just wanted to tell you. And another question from the audience, from Yudong Lee. Thank you, Oscar. Nice talk. One question from my side. Do you think the future it is possible or necessary to use these synthetic AAPCs to achieve T cell activation in vivo? There are a few examples using LNPs, but not with the cell size artificial cells, as far as I know.
Mhm. Uh, the short answer is yes. Yeah. Everything that we use is medical grade. The lipids can be medical grade. The oil it's not organic oil. It's simple silicon oil that you could implant in principle. Yeah. Surfactants that we use are not harmful. The proteins and so on. It's all compatible. Are we pursuing this? No. Yeah, mostly for scientific reasons. We're currently more interested in understanding the mechanisms that drive this regulatory phenotype. Um, would it be clever to do it? I think so, but in a very specific context. So what I could imagine is that you have a synthetic tissue, a synthetic lymph node that you can inject under your skin, probably and you have a reservoir of a specific antigen. For instance, a tumor-associated antigen. Yeah. So in that case, that could serve as an external training hub for immune cells in the vicinity of a tumor, for instance, where they can go pick up some antigens, maybe take a rest, yeah, have a nice environment to reactivate and proliferate before they can go back into a tumor. Yeah, but you see, it's a very advanced therapeutic scenario that I think if you really want to go and compete with what is out there, just the logistics behind it and how much the gain for individual patient is on it, I don't really know if it's worthwhile pursuing at the moment.
Okay, thank you. So Felix is asking, do you see any relationship in between the increase in confinement per se in your dense tissue compared to your loose one regarding the measure cell trajectories? Oh, this word is killing me.
Can you repeat the first part of the question?
Don't make me repeat this word. Do
No,
you
just
see
just
any,
just the first one. Yeah.
do you see any relationship in between the increase in confinement per se in your dense tissue compared to your loose one regarding the measure cell trajectories? Phew. First part of the question.
I'm not sure if I understand very correctly, but so what we see is that the cells migrate differently, yeah, in a dense tissue as compared to loose tissue, which maybe is not too surprising. Yeah, so they need to squeeze through more intracellular channels that are smaller, that just hampers their trajectory. The important thing is that this correlates with their ability to reach a decision whether they have received enough information for being activated or not. Yeah, this is what interests us mostly. Yeah. So how does speed, persistence, and the total trajectory traveled, as well as the dwell time of individual synthetic cells, correlate with their activation on a single cell basis? Yeah.
Okay.
I hope I understand the question correctly.
Yeah, I think so. I think that was the second part of the question. I will read it out loud, nonetheless. I would expect as the increased confinement degree overall cell travel distance, hence impact your interpretation. Okay, I don't even know this word. If possible, as follow up, do you observe similar trajectory in absence of presenting ligands in both tense and loose tissue? And I think that's
Yeah,
what we
exactly.
just commented on.
Yeah. So what we can say is now it makes a little bit more sense. Yeah. The if we can also create just synthetic tissues that don't have any biochemistry inside, so no antibodies, and of course we do that as a control. And you also see that the cells will swirl in. We actually don't know why they like to go into that synthetic tissue if there's no biochemical attractant that makes them go there. But they say they see the same impact. Yeah. So the migration is not driven or mostly decided by the type of biochemical signal. But if you put them there, the dwell time actually increases a little bit. Yeah. So they find a biochemical trigger and they will have a look at it, simply speaking. Yeah. But the migration in between triggers is mostly defined by the synthetic tissue architecture.
Yeah, so maybe mechanics come back. Who knows? Okay, so last two questions, and then we also up with the time. So Mariana asked, great talk and fantastic work. Thinking back to the T cell therapies for patients, how exactly do these synthetic cells versus expanding the patient's own cells would increase the percentage of patients that actually respond to the therapy, increasing that
I think
30%?
I didn't say that. Yeah. So the general approach that we are pursuing with this is quality over quantity. So the dynabeads are really good at producing in a very short amount of time a lot of T cells. Yeah. So these T cells, we are speaking about expansion folds of 10,000, maybe up to 100,000 cells. Yeah, from one cell, 10,000 to 100,000 cell within the order of 1 to 2 weeks. Yeah, so super strong proliferation, very fast. Yeah. So the idea is the more cells, transgenic cells that you put in back into a patient, the more cancer cells you can kill, or the higher chances you have that they will find the tumor and kill the cancer cells. Well, if you look at the final product and the quality of these T cells, you will see high expansion rates also means they're very susceptible to being suppressed, and they're not really good killers at the end. So what we are pursuing is maybe it's worthwhile only achieving 1,000-fold expansion or 100-fold expansion, but increasing the quality of the T cells that we infuse so that every single T cell is actually a good killer, or as we found, maybe also a good regulator at the end. So the idea is making these 70% of the patients benefit by making better T cells, not making a better CAR T cell software behind it.
And follow-up question by me, have you actually thrown your activated T cells on cancer tissue, patient-derived cancer tissue, see what is happening?
Yes, not on patient-derived cancer tissue, but we have used them for bispecific T cell engager therapy. So it's kind of a proxy for, well, it's an approved immunotherapy, but it's a proxy for understanding how good killers they are. And they can kill quite efficiently, and they can regulate even better in that case. Yeah. But we've, for instance, also performed the very same process for tumor infiltrating lymphocytes, which is a special type of immune cell you want to take out of a patient's body.
Okay. Good. Then we're coming to the last question from Kushi Tyagi. Hopefully I pronounced it in any way right. Thank you for the great webinar. Your work involved developing a completely new synthetic tissue platform and integrating concepts from physics, immunology, and engineering. What was the most challenging obstacle that you faced during this project, and what mindset helped you to overcome it when things didn't work as expected?
I feel like someone wants a motivational answer to this question. Yeah. So I need to think about it. Yeah. Because probably that person is putting a lot of weight into my answer now here. Yeah. Gonna give you a very honest answer to it. Yeah. Coming from synthetic cell engineering, from the world of biophysics and chemistry, you know how good your control of a system is, how defined the processes are that you can do. You can quantify thousands of particles. You can exactly calculate and predict their behavior. And then you suddenly start taking T cells from patients and you see biology as all this variation. Yeah. We have patients that simply don't activate at all, neither with our dynabeads nor our tissues. You have patients that activate always very well. You have patients that sometimes activate, sometimes not. So just coping with the degree of variability in biology and in patient population is a real challenge. And honestly, it's more for the PhD students actually investing all the time at the pipettes than just to find out a week later that two of the three donors that they picked just didn't work out. Yeah? Not because the tissue's bad or because the concept doesn't work, but because the patient is as the patient is at the end. Yeah? And how did we cope with this? Well, I can only speak for the PhD students here, I hope, by saying they powered through. Yeah, they were really amazing in testing more and more patients, going back to the lab again, getting new data on this, and understanding if the effects that we observe are real or not.
Okay, thank you. And learning that failure is also part of the PhD, I guess. Troubleshooting is like the main task. So thank you, Oskar, for this great talk and the great
Thanks.
question and answer session. So thank you, everybody, for joining today. And we have now 2 months of summer break, and we will be back in September, I think. If you check our website, you will be informed accordingly. And then thank you again, Oskar. Have a nice afternoon, and to the rest of you too. See you soon.
Thanks. Thanks for having me. Bye bye.
Bye.